Hello, my name is Devin Peart. I'll be talking to you about the metabolic change of lipid species in stem mice with hepatocellular carcinoma. What is hepatocellular carcinoma, shorthand HCC? Well, it's a primary liver cancer which is ranked second for cancer-related death worldwide. And this rate of occurrence is increasing. And you might be asking why. Well, a known risk factor is obesity, which increases the risk 89%. Well, what's the association or relationship between HCC and obesity? Well, it's fat. People today throughout the world are weighing heavier than they have in the past, such as, and that fat or lipids change the liver's microenvironment. So hallmarks for HCC is first it's satosis, which is a fatty liver. And then when that fatty liver develops inflammation, we define that as non-alcoholic steatohepatitis, which then can develop into cirrhosis, which is chronic liver damage with scarring. And then finally we have HCC, or the cancer. And this is how we define the stages of non-alcoholic fatty liver disease induced HCC. And there's actual multiple hit theory on how the fatty liver disease induces HCC. So first, satosis. Well, satosis again is fatty liver, and when the microenvironment is insulin resistant and there's an influx of lipids, such as if you're eating a high fat diet, your liver can go into satosis. And when you add cellular stresses such as oxidative stress and apoptosis with a few gene interactions, that it gives to that multiple hit theory where the microenvironment allows a NASH-induced HCC. So how do you define lipids? Well, it's actually quite hard to define lipids as there's no international body with a definition. And so between scientific articles and Google search, that much of not cannot be credited because they're too different. So we define ours by lipid max. This is a site where people are coming together for lipidomic research, which this is. And so here's the definition, which is actually more of a biochemistry definition, and it's very technical, which is helpful for us as we're using mass spectrometer to identify lipid species. However, other definitions are usually too short and vague. So there's 8 groups for classification of lipids, and we're looking at 3 of them: glycerolipids, glycerophospholipids, and sphingolipids. Again, I'll go over fatty acids because that is structurally important for the other 3. So fatty acids are commonly known as fatty acids. There's going to be 3 different types, which are going to be saturated based on 0 double bonds, monounsaturated, which are 1 double bond, and polyunsaturated, where you're going to have 2 or more double bonds. And from the figure, you can see that the structure generally is a carboxylic acid group with a carbon chain, and then if it's saturated, there's going to be 0 double bonds, and then similar, it's going to be a carbon chain, but there's going to be some kind of double bonds. So glycerolipids, or commonly known as triglycerols, which are we looking at, and their general structure is glycerol attached to 3 fatty acids. Glycerophospholipids are complex glycerolipids, and there's 12 classes of them. And again, you can see that the fatty acids are actually part of that structure. With a glycerophospholipid, generally, is they're going to become kind of phosphorus head group. And then ether-linked lipids are just going to have that structure attached to a glycerophospholipid. Sphingolipids, again, as you can see from the structure, fatty acids are part of the structure. And this is important because we identify that the nature of the lipids, such as if it's saturated or polyunsaturated, actually kind of tells a story on the damage was received by the liver. So what is our purpose? Well, we're trying to characterize the lipidome in liver tissue specimens. So we're trying to identify and quantify lipids in different stages in fatty liver-induced HCC, because we want to be able to determine which of these lipid concentrations are distinguishable between the cancer tissues and the non-tumor tissues. So concentrations are good because we're able to potentially use them for early HCC detection and possible treatment outcomes. So how did we investigate? Well, we used a STAM model. And so from the STAM model, as you can see from the figure, that we have 5 different stages to replicate the fatty liver disease-induced hepatocellular carcinoma. So we have satosis, steatohepatitis, chronic fibrosis, HCC, and also we looked at the non-cancerous tissues of an HCC liver, which is helpful in identifying, can we use lipids to identify the cancer in a liver tissue, in a liver that is the cancerous liver? So can we identify the tumor? And also, so from tissue extraction, we use a mass spectrometer to quantify and identify these lipid species based on their mass to charge ratio. So we took phospholipids and identified them by head group in total acyl carbons and double bonds, and then triglycerols, 2 ways fatty acyl group with some remaining mass that would form the triglycerol, and 3 fatty acyl groups that would create a triglycerol, such as the structure that I showed. So how do we distinguish lipids? Well, first, based on identifying the lipids, we had almost 2,000 lipids identified. However, not all of those are going to be reliable. So those ones with below the limited detection, we consider those unreliable, and we filter those out. Next, we took the concentrations and see if they vary from the internal standards asked during the mass spectrometer analysis. And if they vary too greatly, we filter those ones out. Again, those filtered out ones may be important, but for this purpose, we're trying to reduce the number we have, which this allowed us to get around 200, 250 lipid species to further investigate. And next, so we're trying to distinguish between lipid species. So if the mean concentrations between all the 6 groups were the same, we filter those ones out because we're only looking for ones that are distinguishable or have mean concentration differences. And so the next, we used a piecewise comparison to determine which 2 groups had a lipid species mean concentration difference. So quickly, so we have phospholipids that were distinguishable between the HCC tissues and the non-cancerous tissues of an HCC liver. For the study, they're 2 separate tissues. However, from mice, we're comparing them as, can we identify the tumor in a cancerous liver? And phospholipids actually decrease, while triglycerols we can see increase. This is the triglycerols with an actual and some mass to form my triglycerol. And the highest one we have here is 87% mean concentration difference. And again, triglycerols with 3 acyl groups, we have 134% mean concentration difference higher in HCC tissues. So our conclusion is that we're able to distinguish between tumor and non-tumor tissues, such as that hepatocellular carcinoma and non-cancerous HCC liver tissues are distinguishable by the 15 mean concentrations we found, and they should be considered as potential early detection biomarkers or treatment options for HCC. And that we found that actually the nature, such as saturated and monosaturated nature lipids, were higher in the concentrations in HCC, which may explain the origin of liver damage needed for HCC development. Reminder, fatty acids may be good or bad, and we saw more of those bad ones in the HCC tissues. So here's my future work and limitations. Knowledge, thank you, Kansas Research Center as they did the extraction and analysis of the lipid profiles and my references. Thank you and have a good day.